Hi, and welcome to the course on Next Generation DNA Sequencing Analysis. In this video, we will explore the applications of next generation sequencing. The DNA double helix has two strands. Each strand consists of subunits called nucleotides linked together. These nucleotides come in four flavors: A, T, C, and G. The sequence of nucleotides in our DNA carries the genetic code. DNA sequencing involves finding the sequence of letters. All the DNA in one of our cells is called our genome. Sequencing the first human genome took 13 years and $3 billion. We can now sequence an entire human genome in just 1 or 2 days for $1,000. This improvement was possible after a new sequencing technology, next-generation sequencing, was introduced in 2007, just 4 years after the first human genome was sequenced. Next-generation sequencing, or NGS, provides a much higher sequencing capacity at a lower cost. This has made it feasible to sequence many genomes. So what? What can we do with many genomes? We can compare the genomes of hundreds or thousands of people with and without an inherited disease. We can find the genetic markers that are found only in people with the disease. These markers can help us find the genetic cause of the disease. This helps us understand the disease and develop better prevention and treatment. Personalized medicine is now possible, where we treat patients according to their genes. Many drugs work well on most people but cause severe side effects for a small proportion of the population. Armed with a DNA sequence of a patient, we can predict the patient's response to a drug before prescribing it. We can select the drugs that will be most effective for them while avoiding severe side effects. We can sequence the DNA of cancer cells and find the mutations that are causing it. We can then prescribe drugs that target these specific mutations. Humans have been selecting crops and livestock based on desired traits like high yield or climate resistance for hundreds of years. We can now compare the sequences of many individual plants or animals to discover the genetic markers of desired traits. This lets us choose plants or animals for breeding based on a combination of desired genetic markers. This is more precise than traditional selective breeding and leads to faster improvements in the crop variety or cattle breed. We often culture microorganisms to produce a product, like human insulin, which is produced by a culture of genetically modified bacteria. Since microbes multiply rapidly, genetic variation can arise quickly. If genetic differences accumulate in our culture, the microbes may no longer be producing the correct product. We can sequence the DNA of the culture to ensure that it is genetically stable and is producing the product we expect.